Swayam Prabha. Digital India. Educated India.
Hello learners, I am Dr. Rekha Sharma, retired professor UGC Human Resource Development Center, Rashtrasant Tukadoji Maharaj, Nagpur University, Nagpur. In this video, we shall study classification of lipids and types of fatty acids. Now the learning objectives of this module. After studying this module, the learners will be able to classify lipids or fats. They will be able to describe different types of fatty acids. Students will be able to state essential fatty acids and explain trans fatty acids. Now, what are lipids? Lipids are a broad group of chemically diverse compounds that are soluble in organic solvents like ether, chloroform and benzene. Lipids which are solid at room temperature are referred to as fats, and those which are liquid are oils. Food lipids are also classified as non-polar, for example, triacylglycerol and cholesterol, and polar lipids, for example, phospholipids, to indicate differences in their solubility and functional properties. Polar lipids often contain a hydrophilic head group that has a high affinity for water attached to a lyophilic tail group that has a high affinity for oil. Now, chemical composition of fats. Fats are composed of glycerol and fatty acids linked together to form ester. 1, 2 or 3 fatty acids may be esterified with glycerol to form monoglycerides, diglycerides or triglycerides respectively. The most common form of food fats are triglycerides or triacylglycerols. Triglycerides are made up of 3 fatty acids esterified to the glycerol molecule. Now let us try to understand the chemical composition of fats. Here you can see that one molecule of glycerol is attached with 3 fatty acids chain, and the resultant product is triglyceride or neutral fat. Now it will be a neutral fat if all these 3 fatty acids are same plus 3 water molecules. So, one molecule of glycerol combines with 3 fatty acids and forms triglycerides or triacylglycerols plus water. So, we will try to understand this. This is a molecule of glycerol. It is made up of CH2OH, CHOH, CH2OH. Now, when it combines with one fatty acid, this is one fatty acid chain. It is one end of fatty acid has COOH group and the another end has CH2 group. So, this is one fatty acid, this is another fatty acid and this is the third fatty acid. When these 3 fatty acids they combine with the molecule of glycerol, let us see what happens. Now, this glycerol, now it has only CHOH, 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 and when it combines with one fatty acid, it is known as monoglyceride. This is known as diglyceride, and when 3 combines, it is known as triglyceride. Now, along with the formation of triglyceride, there is a formation of water also. So, 3 molecules of water are also formed. Now, classification of lipids. Lipids are broadly classified into 2 groups: simple lipids and compound lipids. Now, simple lipids. Simple lipids include fatty acids fats which are esters of fatty acids with glycerol, for example fats and oils. Waxes are also simple lipids. They are esters of fatty acids with long chain monohydroxy alcohols or alcohol other than glycerol. This is an example. Wax is an ester of a long chain acid and a long chain alcohol. In reality, industrial and food waxes are a combination of chemical classes including wax esters, sterol esters, ketones, aldehydes, alcohols, hydrocarbons and sterols. Waxes can be classified according to their origin as animal, that is beeswax, plant, that is carnauba wax, and mineral, that is petroleum waxes. Waxes are found on the surface of plant and animal tissues to inhibit water loss or to repel water. Waxes are commonly added to the surface of fruit to slow dehydration during storage. Now, compound lipids. Compound lipids are defined as esters that contain chemical groups in addition to fatty acids and alcohol. Depending on their chemical groups, they are further subdivided into glycerophospholipids, sphingolipids, glycolipids, sulfolipids, lipoproteins. Now, what is phospholipids? The phospholipids or phosphoglycerides are modifications of triacylglycerols where phosphate groups are typically found in the SN3 position. The simplest phospholipid is phosphatidic acid PA, where the substitution group on the phosphate at SN3 is an OH. Other modifications of the substitution group on the phosphate at SN3 result in phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine and phosphatidylinositol. Now, sphingolipids. The sphingolipids are lipids that commonly contain a sphingosine base. Common sphingolipids include sphingomyelin, ceramides, cerebrosides and gangliosides. Now, sterols. They are not made up of fatty acids and glycerol but have a benzene ring structure. These fat-like substances include cholesterol and fat-soluble vitamins, vitamin A, D and K. Sterols are derivatives of steroids. These non-polar lipids all have 3 6-carbon rings and a 5-carbon ring that is attached to an aliphatic chain. Sterols have a hydroxyl group attached to carbon 3 of A ring. Sterols are found in both plants, phytosterols, and animals, that is zoosterols. Cholesterol is the major sterol found in animal lipids. Now, fatty acids. The major components of lipids are fatty acids, compounds that contain an aliphatic chain with a carboxylic acid group. Fatty acids with a odd carbon numbers and branched chains can be found in sources such as microorganisms and dairy fats. Fatty acids are carbon chains with a methyl group, that is CH3 group at one end of the molecules designated omega azo and a carboxyl group COOH at the other end. The general formula of fatty acid is CH3 CH2 n times COOH, where n varies from 2 to 22. The majority of fatty acids in nature range from 14 to 24 carbons. Some fats contain fatty acids with less than 14 carbons. Significant levels of short-chain fatty acids are mainly found in tropical oils and dairy fats. Now, classification of fatty acids according to the length. Short-chain fatty acids have less than 6 carbon chain length, that is smaller than caproic acid. Butter contains small chain fatty acids. They are also obtained during fermentation. Medium chain fatty acids have 6 to 10 carbon chain length. They are present in butter and coconut oil. Long chain fatty acids contain more than 12 carbon chain. Palmitic acid and stearic acid are major fatty acids present in diet. Now, classification of fatty acids according to degree of saturation. Saturated fatty acids have single bonds between carbon atoms. Unsaturated fatty acids have one or more double bonds between the carbon atoms. Triglycerides from animal sources contain a higher percentage of saturated fatty acids and are normally solids at room temperature known as fats. The plant triglycerides are rich in unsaturated fatty acids, are generally liquids at room temperature and are called oils. Now, saturated fatty acids. Saturated fatty acids are filled or they are saturated with hydrogen. Examples of saturated fatty acids are butyric acid, which is present in butter, stearic acid found in animal fat and plant oil, lauric acid found in palm kernel and cinnamon. Now, unsaturated fatty acids. Unsaturated fatty acids have one or more double bonds. Fatty acids having a single double bond are called monounsaturated fatty acids or MUFA, whereas those having two or more double bonds are called polyunsaturated fatty acids or PUFA. Now, unsaturated fatty acids are differing with respect to the number of carbon atoms and double bond characteristics are found in lipids. Unsaturated acids may have 1 to 6 double bonds. Double bonds occur in many locations, but a regularity of position is seen in most of the acids commonly found in food fats. Now, nomenclature of saturated fatty acids. Fatty acids can be described by systematic, common and abbreviated names. The IUPAC system names the parent hydrocarbon of the fatty acid on the basis of the number of carbons. For example, 10 carbons would be decane. Since fatty acids contain a carboxylic acid group, the terminal E in the hydrocarbon's name is replaced by oic, like decanoic. Now, many of the common names originated from the source that the fatty acid was commonly or traditionally isolated, for example, palmitic acid and palm oil. A numerical system can be used for abbreviated names. The first number in this system designates the number of carbons in the fatty acids, while the second number designates the number of double bonds. For example, hexadecanoic, that is palmitic, and it is represented as 16, 0. Obviously, the second number will always be 0 for saturated fatty acids. Fatty acids that contain double bonds in their aliphatic chain are referred to as unsaturated fatty acids. In the IUPAC system, the anoic, that is ANOIC designation is changed to enoic, enoic to designate the presence of a double bond. On the basis of the number of double bonds, the terms di, tri, tetra and so on are added. Common names also exist for the unsaturated fatty acids, with the exception of some of the long chain polyunsaturated fatty acids, and the numerical abbreviation system is similar to the saturated fatty acids, with the second number indicating the number of double bonds. For example, octadecadenoic is represented as 18s2. The positions of the double bonds in the IUPAC system are numbered by the delta system, which indicates the position of the double bonds from the carboxylic acid end of the fatty acids. For example, oleic acid, which has 18 carbons and 1 double bond, would be 9-octadecanoic acid, and linoleic acid, which has 18 carbons and 2 double bonds, would be 9-12-octadecanoic acid. An alternative numbering system that indicates the position of the double bonds from the methyl end of the fatty acids is known as the omega system, sometimes given a shorthand notation of N. Now, polyunsaturated fatty acids or PUFA. Polyunsaturated fatty acids include linoleic acid with 2 double bonds, linolenic acid with 3 double bonds, and arachidonic acid or arachidonic acid with 4 double bonds. Now, in PUFAs, the first double bond may be found between the third and fourth carbon atoms from N carbon. These are called as N3 or omega-3 fatty acids. If the first double bond is between the sixth and seventh carbon atoms, then they are called N6 fatty acids. The double bonds in PUFAs are separated from each other by a methylene grouping. Now, cis and trans fatty acids. The natural configuration of double bonds in unsaturated fatty acids is the cis configuration. In the cis configuration, the carbons of the aliphatic chain are on the same side of the double bond, while trans double bonds would have the carbons on opposite sides. Double bonds in the cis configuration will cause the fatty acid to arrange in a bent configuration. Here you can see. Thus, unsaturated fatty acids are not linear, making it difficult for them to orient themselves into tight packing configurations. Trans isomers may be produced during industrial processing, for example, hydrogenation of unsaturated oils, and in the gastrointestinal tract of ruminants. The presence of a double bond causes a restriction in the mobility of the acyl chain at that point. The cis fatty acids are thermodynamically less stable than the trans forms. The cis fatty acids have lower melting points than the trans fatty acids or their saturated counterparts. Now, essential fatty acids. 2 of the polyunsaturated fatty acids cannot be synthesized by the body and have to be provided in the required amounts by our diet and are called essential fatty acids. They are linoleic acid, that is omega-6 fatty acid, and linolenic acid, that is omega-3 fatty acid. Now, vegetable oils and animal fats. Most vegetable oils, especially those from oilseeds, are highly unsaturated and contain primarily fatty acids in the 18 carbon series. Oils high in oleic acid include olive and canola. Oils high in linoleic include soybean and corn, and oils high in linolenic include linseed. Triacyl glycerols from plant sources that contain high amounts of saturated fatty acids include cocoa butter and tropical oils, for example, coconut. Coconut and palm kernel oils are also unique in that they contain high amounts of medium chain fatty acids 8s0 to 14s0 with 12s0 predominating. Now, animal fats. The level of saturated fatty acids in fats and oils from animals is generally in the order of milk fat greater than sheep, greater than beef, greater than pig, greater than chicken, greater than turkey, greater than marine fish, with palmitic and stearic being the major saturated fatty acids. The fatty acid composition of animal fats is dependent on the digestive system of the animal, with fat from non-ruminants, for example poultry, pigs and fish, being partially dependent on the fatty acid compositions of their diets. An example of this is pigs such as Siberian hams, where dietary regimes are manipulated to produce lard with a high oleic acid composition. Among the non-ruminants, triacylglycerols from marine animals are unique because they contain high amounts of the omega-3 fatty acids, eicosapentaenoic and docosahexaenoic acid. In sheep and cows, dietary fatty acids are subject to biohydrogenation by microbial enzymes in the rumen. This results in the conversion of unsaturated fatty acids into saturated fatty acids and can also produce fatty acids with conjugated double bonds such as conjugated linoleic acid, CLA. Since ruminants consume primarily lipids of plant origin, where the fatty acids are primarily in the 18 carbon series, the end product of this biohydrogenation pathway is stearic acid. Thus, butter, beef fat and sheep fat contain higher amounts of stearic acid than fats from non-ruminants. Ruminal bacteria are also unique in that they can ferment carbohydrates to acetate and beta hydroxybutyrate. In the mammary gland, these substrates are converted to fatty acids to give butter fat a high concentration of saturated short chain fatty acids that are not found in other food triacylglycerols. Ruminal bacteria also promote the formation of keto, hydroxyl and branched fatty acids. Because of the impact of ruminal bacteria on fatty acids, butter fat contains hundreds of different fatty acids. Now let us summarize. Fats, oils and fat-like substances belong to a group of substances collectively known as lipids. Lipids are classified as simple lipids and compound lipids. Simple lipids are made up of one molecule of glycerol and three molecules of fatty acids. Compound lipids are esters that contain chemical groups in addition to fatty acids and alcohols. Fatty acids may be saturated, monounsaturated or polyunsaturated. Depending on whether saturated or unsaturated fatty acids are present, the fat is solid or liquid at room temperature. Two polyunsaturated fatty acids, namely linoleic and linolenic acid, are essential as they cannot be synthesized by the body and must be present in the diet. In the cis configuration, the carbons of aliphatic chain are on the same side of the double bond, while trans double bonds would have the carbons on opposite sides. Most vegetable oils, especially those from oilseeds, are highly unsaturated and contain primarily fatty acids in 18 carbon series. The level of saturated fatty acids in fats and oils from animals is generally in the order of milk fat greater than sheep, greater than beef, greater than pig, greater than chicken, greater than turkey, greater than marine fish, with palmitic acid and stearic being the major saturated fatty acids. These are the references. With this, we have come to an end of the lecture on classification of lipids and types of fatty acids. Thank you.